Hello everyone, good morning. We are going to start another lecture. So, our today's topic is Biomedical Lab Techniques 1 Introduction. Why this is important? Because we have to learn in upcoming lectures cell culture, microscopy, PCR, flow cytometry, ELISA, and Western blot. And all of these techniques need a scientific question. A structure inquiry that utilizes acceptable scientific methodology to solve problems and create new knowledge that is generally applicable. So, simple question relating to day-to-day activities employed to formulate interactive theories or law that govern our lives. And difference lies in the degree of formality, rigor, verifiability, and journal validity. Scientific methods are depends on ask a question, do background research, construct a hypothesis, test with an experiment, analyze results, draw a conclusion. If hypothesis is true, report results. If hypothesis is false or partially true, then think again and then construct hypothesis again and repeat the same cycle. So simple question yes or no with answer or maybe or a value. Background researchers have the same or similar question been asked previously. If this, then that happens. Must be testable. Appropriate experimental method change one variable at a time. Our result represent the scientific question asked or due to the change. And then discuss with colleagues, friend, and your mentors. So, what are the characteristics and requirements of scientific research? Scientific research is control. In exploring causality in relation to two variables, you set up your study in such a way to minimize the effects of other factors. For example, experimental drug. A value or experimental drug A will cure disease B, sex, age, ethnicity, route of delivery, dose, disease progression. It can be rigorous research methods used are reliable, appropriate, and justified, or can be systematic. Research methods use follow a certain logic sequence. Some procedure must follow others. So valid and verifiable uh, are conclusions based on findings are correct. Findings can be verified by you and others. Empirical conclusions draw are based upon hard evidence collected from real life experiences or observations. And they can be critical process adapted and procedures used must be able to withstand and, and critical scrutiny. So the reality of scientific research is science is complex, iterative, and can take many different paths. Involves many different people engaged in different sorts of activities in different order and at different points in time. It is a dynamic, flexible, and unpredictable process. This course will share this characteristic, but don't be panic. You will not be penalized to for a failed experiment as long as you follow the instruction I provide. So for scientific research, lab notebook play a crucial role. The single most important piece of equipment you will use when working in lab. Science is built upon the premise that results are reproducible, must leave detailed information so others can reproduce your work. Notebook should be an accurate account of what you did, why you did it, when you did it, what the results were, and what these results mean. Good science require good record keeping. The importance of lab notebook is record of progress of your experiments. 1 week, 1 month, 1 year, decades. Houses lab protocols, publications and ground records, list of future plans and ideas worth exploring. Effective teamwork helps you stay organized. Practice or GLP is a code of federal regulation which is called 21 CFR Part 50 FDA. Excuse me. This became effective in June 1979, and this code established a standard for the conduct and reporting of non-clinical laboratory studies. This is intended to ensure the quality and integrity of safety data submitted to FDA. Documentation of research ensure work is done according to agreed procedures and that data is properly recorded. Laboratory's notebook is legal document. Your data may have to be explained, defended, reconstructed, or repeated without your assistance, so others must be able to understand what you did during your research experimental work or research studies. Notebook, also called intellectual property. US patent law states that inventorship is determined by the first to invent, not the first to file. Dates of conception and reduction to practice. Successful testing of your idea are key. During conflicts over a patent, each party has an opportunity to submit documentary proof. A sketch and brief written description are usually sufficient to establish conception. In many legal cases, laboratory notebook has been the crucial piece of evidence to justify who is the owner of the particular patent. So, in this example of intellectual property case, Dr. Tunhu Lee discovered GB120, the glycoprotein used in HIV/AIDS test, while working in the Essex labs. His notebook helped all lawyers successfully defend the GB120 patent. So there are certain record keeping guidelines. So we should make a table of content. Leave the first 2 to 4 pages of the notebook blank. Record only the first page number of each multi-page experiment. Quickly find information you seek. When is data recorded? Entries should be made as the experimental process proceeds, but no later than as soon as it is done or when an idea is conceived. Try to avoid writing on post-it notes or scraps of paper and transcribing into your notebook later. Each page is numbered and dated. Each recorded lab should have the following parts. Title, be specific and concise. Objectives or purpose of the lab. Concise statement about or definition of the experiment. Plan, outline, or flow diagram of lab. This can be optional and a step-by-step procedure. Simple sentences stating the work that was done. Raw data, results including graphs, tables, figures, photos, and drawings. Each recorded lab should have the following parts: conclusions, including the biological and chemical concepts involved, whether the objectives was met, any problems encountered, and suggestion for the next step and or future experiments. Critical information to include. What are some examples of critical information one would include in their notebook? Concerning solution, it is important to note names be specific, not just the buffer. Lot number, variability between batches exist. List the purity, concentration, etc. List the source, catalog number, record your recipes. related to the reconstructability and repeatability of the experiment. Other things can also be recorded, notes of unexpected results or observations, any deviations to the normal protocols, mean like written protocols say use 1 millimolar sodium chloride, instead you use zero point 0.5 millimolar sodium chloride, so you have to make this as a deviation. All measurements and important test conditions, weighs, volumes, temperature, times, should be noted. All units, gram, milligram, microliter, molar, nanomolar, everything should be noted. If you could make a diagram of one part of the procedure section of lab notebook, that will be very helpful. So it will be useful when setting up a piece of equipment, or must be understandable and clearly labeled. Also a recording of what you observed. Result section should comprise on written results. Data must be clear, understandable, and complete. Write down everything. Don't be embarrassed about writing down mistakes or accidents. Is your data legible to others? Is your data clear, detailed enough that someone else in your discipline could understand it and repeat it? Are abbreviations defined and obvious? Have you included drawings and flowcharts to improve clarity? Notebooks can also contain glued-in information, printed graphs, data sheets, photographs, machine printouts, especially for gel images. Items can be taped, glued, or placed in protective sleeves. Keep location consistent with the written notebook. Discussion and conclusion. This section must have summary of your work, what was done and what was learned, what went right and what, if anything, went wrong. New ideas should be recorded here. How could we improve this experiment? Any idea may occur only once and only briefly. Catch it while you can and record it onto the page. So conclusions can also contain fact versus opinion. Remember that science is objective and unbiased. There are fact expected results were not obtained versus opinion the experiment was not good. No reaction was observed versus opinion there these two chemicals don't react. Another fact, under these circumstances the reaction was unsuccessful. Opinion, the experiment was failed. Mistakes, never use whiteout, never erase anything in notebook, never write over, never discard or replace attached supplementary data. Always record a defensible reason for the correction added. Always circle the reason or draw a single line through. Always add your dated initials to the corrected edited data after the circle reason. So what not to do on a lab notebook, never remove a page. Gaps suggest missing data. Do not use pencil or ink that easily smears. Do not include notes information that are irrelevant to your experiment. No rude or offensive comments. Avoid subjective statements or opinions. Only facts, objective measurements, and statements reported by the data. Do not remove the notebook from the lab. So, what is wrong with this notebook entry? So, here we can see there is no heading, no date, no initials. Task 1 without heading, task 2 without heading. On the result part, values for T was missing, and also chart title was also missing. So this is not good for recording your results in notebook. So the legend of diagram are following 20 points. I'm not going to read all of those points, but we can clearly see from number 1 we have continued from page 3, page number, project title, page 4, and this is June 3rd, the date here. Title number 3 is described laboratory abbreviations and designations are defined and referred, and so on. By end of the page number 20 point, each page is labeled and confidential. Here we can see there is a brief description of the result. This is sp2 probe versus FET, made out of the sample blah blah by pulling it again. So that's how the results are recorded with this description. And conclusion also noted. Nowadays, this is easy to record this result section in file of Word. But so many people now like to use electronic laboratory notebooks, also called ELNS, and they are growing in popularity, especially Microsoft Word, dedicated software, and iPads. Search and organization functions, links to analytical software, on-site storage, or also on cloud, so you can access anywhere. So here we can see different type of lab notebooks. So bound or stitch, no lost pages, legally strong. Disadvantage is difficult to copy, not logically organized data might be stored elsewhere. Another type is loose leaf binder. Advantage is easier to organize by experiment, data can be stored together. But disadvantage is sheets can be lost, difficult to authenticate dates. Computer electronic type, easy to search, easy to read, easy to share with collaborators, large data storage capacity. Disadvantages requires electronic security, files can become corrupted, software compatibility issues. Where and how long to keep notebook? In progress, progress notebook should be kept in the lab and reviewed periodically. Completed notebook should be indexed, catalogued, and kept in a safe place. Original notebook should never leave in the lab. Individuals may be permitted to take copies of their notebook with them when they leave. Keep for at least 5 years after funding for a study ends or for the life of the patent plus 6 years. That is all for today's introduction to lab, and first part was lab notebook. If you have any question, please reach out to me via comment. I will be happy to answer your question. Thank you for watching today's lecture, and we'll see you soon again. Have a good day. Bye bye now.